Welcome to one of the most fascinating mysteries in evolutionary biology. Today, we are going to explore the Dipleurula concept. Imagine finding a heavily armored knight and a swift Olympic runner, and discovering they are brothers by looking at their identical baby pictures. We will see how this exact scenario plays out between starfish and humans, revealing our deepest evolutionary origins. Now that we know what we are looking for, let us talk about these evolutionary baby pictures. In the animal kingdom, an adult starfish, which is an echinoderm, and a human being, a chordate, look absolutely nothing alike. However, when we examine their microscopic, free-swimming larval stages, they share a striking, undeniable resemblance. This incredible similarity led scientists to a profound realization. Both of these vastly different groups must have evolved from a single shared ancient ancestor. We affectionately call this ancient ancestor the Dipleurula. Building on this shared ancestry, we must formally define what this creature actually was. The Dipleurula is a hypothetical, extinct, microscopic marine organism. When we say hypothetical, it means we have not found a physical fossil of this exact creature yet. Because it was soft-bodied and microscopic, it did not leave behind a traditional fossil record. Instead, its existence is strongly deduced from the shared developmental traits of its living descendants. Scientists believe it is the universal common ancestor of all echinoderms, hemichordates, and chordates. Since we cannot look at a fossil, let us reconstruct what this ancestor looked like based on its descendants. This ancestral creature was bilaterally symmetrical, meaning its left and right sides were perfect mirror images. This is fascinating because adult starfish have a circular radial symmetry, but their origins are bilateral. It possessed a simple digestive tract with a distinct mouth and an anus. To survive in the ancient oceans, it gathered food using a specialized band of tiny hair-like structures called cilia, filtering nutrients directly from the water. Beyond its physical shape, there is a deeper developmental secret that ties us to this creature. Most importantly, the Dipleurula was a deuterostome. This is a highly specific biological trait that occurs during embryonic development. In deuterostomes, the very first opening to form in the embryo becomes the anus, while the mouth forms second. This might sound like a minor detail, but it is a massive evolutionary marker. It is a highly specific trait shared by you, me, and the starfish, proving our shared lineage. To truly appreciate this connection, let us look at the biological evidence side by side. Here, we see a comparative diagram of three larval forms. On the left is the hypothetical Dipleurula. In the middle is the bipennaria larva of an echinoderm, like a starfish. On the right is the tornaria larva of a hemichordate, like an acorn worm. Notice the striking morphological similarities. Look at the continuous ciliated bands looping around their bodies, the identical gut structure with a distinct mouth and anus, and the overall body plan. These shared features across all three forms strongly suggest a common evolutionary origin. Seeing these similarities naturally leads us to ask why this tiny, extinct larva is so important to science. The Dipleurula concept is absolutely crucial because it solves the massive evolutionary puzzle of where chordates, and ultimately vertebrates like us, came from. Before this concept, scientists were completely baffled by how complex animals with backbones could evolve from simpler invertebrate ancestors. The Dipleurula provides the missing evolutionary bridge. It proves that our deepest origins are tied to simple, filter-feeding marine organisms rather than insects or worms, forming the foundation of the echinoderm theory of chordate origin. Now that we understand the biological importance of the Dipleurula, let us explore how we can determine when it lived. Because we lack fossils, evolutionary biologists use a mathematical tool called a molecular clock. This relies on the fact that DNA mutates at a relatively constant rate over millions of years. The formula is T equals D divided by 2 times R. Here, T represents the time elapsed since the two species shared their last common ancestor. D represents the total genetic divergence, or the difference in their DNA. Finally, R represents the genetic mutation rate. We divide by 2 because both lineages have been mutating independently since they split. If genetic divergence increases, the time since the ancestor existed will be much larger. Let us put this formula into practice with a real-world evolutionary calculation. Suppose a geneticist sequences a gene in a modern starfish and an acorn worm. They discover the genetic divergence is 0.54, meaning 54% of the gene is different. The mutation rate is 0.0005 mutations per million years. We need to calculate the time since they shared the Dipleurula ancestor. We substitute 0.54 into the numerator. In the denominator, we multiply the mutation rate by 2, giving 0.001. Dividing these gives us 540. This profound result tells us the Dipleurula lived approximately 540 million years ago, perfectly aligning with the Cambrian explosion. Having successfully calculated the age of the ancestor, let us look at the problem from the opposite direction. What if we find a fossil and want to predict the genetic divergence? To do this, we must rearrange our molecular clock formula algebraically. We start with our original equation, where time equals divergence divided by twice the mutation rate. To isolate the genetic divergence, we need to eliminate the fraction. We do this by multiplying both sides of the equation by the denominator, which is 2 times the mutation rate. This is the first crucial step in shifting our mathematical perspective to solve for a new unknown variable. Continuing from our previous step, we can now finalize our new equation. By multiplying both sides by the denominator, we find that the total genetic divergence, D, is equal to 2 times the mutation rate, R, multiplied by the total time, T. This rearranged formula reveals a direct linear relationship. It tells us that the expected genetic change is simply the combined mutation rate of both lineages multiplied by the total time they have been separated. This predictive power is incredibly useful in evolutionary biology, allowing scientists to know exactly what to look for before they even sequence the DNA in the laboratory. Now that we have derived our predictive formula, let us use it. Imagine paleontologists discover a trace fossil suggesting the Dipleurula lived 600 million years ago. The mutation rate is 0.0004. What is the expected genetic divergence between a modern sea urchin and a lancelet? We substitute our known values into our rearranged equation. We multiply 2 by the mutation rate of 0.0004, and then multiply by the time, 600 million years. The arithmetic gives us 0.48. This means we expect a 48% genetic divergence. If laboratory sequencing matches this, it beautifully bridges the gap between paleontology and genetics. We have covered a tremendous amount of ground today, so let us review our key takeaways. First, the Dipleurula concept is the ultimate evolutionary bridge, connecting simple marine invertebrates to complex chordates through a shared hypothetical larval ancestor. Second, by recognizing shared developmental traits like deuterostome development and ciliated bands, scientists have successfully mapped the origins of our own phylum. Finally, we learned how to use molecular clocks to mathematically verify these biological timelines, calculating both divergence times and expected genetic changes. You now have a complete, masterful understanding of the Dipleurula concept and its vital role in evolutionary biology. Excellent work.